So it's better for us to learn what the barn does and work our vaccine design around the barn. And then the uptake would be, I believe, a lot better appreciated and hopefully used more successfully. So.
Hello, everyone. I'm Dan Columbus, your host for today's Swine Canada podcast. With me today, I have Dr. Heather Wilson, who is a scientist at the Vaccine and Infectious Disease Organization in Saskatoon. How are you today, Heather?
I'm doing well. How are you doing?
I'm fantastic. Thank you very much for joining us today. Heather, just because some of our audience may not be familiar with you and even maybe with VIDO, can we just ask you to maybe introduce yourself, a little bit of your background and how you got to where you are before we get to the topic of the day?
Sure. So I'm Heather Wilson. I got my schooling at the University of Saskatchewan, and then I did my PhD at the U of S in the Department of Biochemistry. I moved over to VIDO, which at the time was called the Veterinary Infectious Disease Organization. It's now been updated to the Vaccine and Infectious Disease Organization. I moved there as a postdoc, and then I started on there as a research scientist, number one back in the day, and I'm been there, I think it's been 16 years now. It's hard to believe it's been that long. So started off with just a tiny little lab, and it gets a little bit bigger all the time. So lots of work with vaccine development and vaccine formulation.
Yeah, it's funny how fast time goes by, and we tend to be victims of our own success. That things get bigger and bigger, and we get busier and busier the more we do. But yeah, I think that's great.
Start doing the math. It's scary, but
yeah. So I know that a lot of your work is involved in vaccine development, and I think this is a big thing, with swine specifically lately, with African swine fever all in the news, and PAD before that. So how about just give the audience an idea of what you've been working on and finding?
Sure. So I'm actually a program manager for kind of like a unit called the vaccine formulation and delivery. So what our group kind of does is try to find platforms for how to make. So you're not specifically disease targeting; it's more generally how to get vaccines to work better. So what that would entail is looking at different proteins to put on the surface, and then finding the spots on the protein that are called epitopes that we should be targeting, and then all the different ways to do that, to find those. If it was easy, it'd be done already, right? So there's lots of development there. So that's called antigen selection. And then there's formulation. So that's the different adjuvants that you put in. So it's the gasoline that gets the car to go kind of idea, that it helps direct how the immune response goes. So sometimes it's better to have an antibody response. Sometimes it's better to have a cytokine response. There's different flavors of the immune response. Sometimes it's always probably a good idea to have vaccines that target the mucosal sites. So if you don't notice that most vaccines, or quite a few of them, are intramuscular injection, which is great because you get a nice immune response, but it doesn't always target the nose, the mouth, the reproductive tract, the digestive tract, and that's where a lot of our vaccines where pathogens gain entry into the body. So if we could find ways to better direct the vaccine response to be at those sites, or anywhere where there's mucus, right? Anywhere on your body like that, you can block the pathogen before it really gets in. The other way works too, like an intramuscular injection or any kind of injection. What that does is it waits for the pathogen to get in, and then it fights. So that's kind of different formulations, and then actually different routes of immunization. So one of the more recent things I'm doing is called intrauterine immunization. And what this is I had a postdoc in my lab a couple of years ago named Dr. Alex Pasternak, who's now at Purdue. And the idea actually came from him, and then we kept working on it and perfecting it. And the idea is that the gilts and the sows are temporarily immobilized when they're being bred, and it's a time when they're already being handled. So the idea was, well, could we actually vaccinate into the uterus during breeding, so with the semen, and have a very good immune response in the uterus and the uterine horns, and protect against reproductive diseases, and actually protect. So if you immunize, then the mom would have antibodies in the colostrum and the milk. So could we protect the neonates, the babies that way as well? So different routes of immunization, and that's kind of a major thing I'm doing right now is this intrauterine immunization in the pig.
Yeah, I think your intrauterine work is quite interesting. But you said something previous. So before we get to that, you mentioned that delivering this for mucosal would be a better option. So why is that not done currently, or I guess like what's in the way, or what do we need to know?
Uh, we do a little bit. If you look, like some of the vaccines, like Intrauterine for Lawsonia intertellularis, like some of them are oral delivery, so it goes into the drinking water. Uh, the trouble there is that your body has spent its entire evolution actively ignoring what you eat and not freaking out and making an immune response to that, and which is great because then it stops you from having dust, a vaccine type response to dust or food or anything that you drink. So, and that's great, except for that's also where some pathogens do get in. So, yeah, anything, the best parts of the body is producing that mucus and having the cilia flush the mucus and the particles out. You've got this really low pH in the stomach, which is killing everything, except for the odd thing that happens to get through. So it's really tough to make vaccines to overcome those harsh barriers. But there are some, certainly. I think all of the ones that I think I'm aware of are all live attenuated. So like in Terrasol, one of the polio vaccines is live attenuated. Like anything that does tend to be delivered to mucosal sites, so intranasal or oral, they tend to be live attenuated. So trying to get the parts of the virus or the bacteria that are present anyway to kind of help move across the gut wall barrier, for example. What we're doing is, it would be nicer if you could actually develop a subunit vaccine that could get across the wall, and what that is playing with the formulation. So we can get it to work. We just can't personally can get it to work, but not get it to work fantastically well. So it just deals with playing with the adjuvants, playing with the timing. And you think too, if you're injecting into the muscle, you're injecting this much, right? If you're injecting into the digestive tract, you're thinking about feet after feet after feet of this small and large intestine, right? So it's also a volume issue. So there's lots of fun things that you have to overcome. But the more we learn about the immunology and the physiology of the animals, the more ideas we can come up with. And certainly it would be ideal if we could just administer a vaccine into feed or into water, and you wouldn't have to have special times where you round up the animals, and you don't have to have more people, more training, cold storage, like all those things. So that is the goal, is to I would think is a major goal, especially for livestock, is putting vaccine into feed or the drinking water.
Yeah, I'm sure the producers would love it if they could put everything through water or something like that. You don't have needle issues and all the things that come with the way that they're traditionally getting.
You don't damage the meat, all that kind of stuff too. So
exactly. So hopefully, could get more of it that way and do that. I guess that leads very well into the work that you're doing in the sows and the intravaginal vaccine. So I've heard some of the stuff that you're doing, but obviously the audience hasn't. So how about just maybe go a little bit more into where you're going with that?
Sure. I'll just correct you there. You said intravaginal. That's another style we're trying to do, but this one actually gets through the cervix into the uterus. So,
Oh, okay.
so that uterine horn. So yeah, well, as your producers probably know, when you inseminate, you either do post cervical, where the actual catheter goes into the cervix and gets kind of pushed through. Most times, it's just the catheter goes halfway in between the cervix, and then what do you have, like 60 or 80 mils of semen. So the idea is right before you're putting on that little tube of toothpaste size semen bag onto the catheter, you would just squirt in a little bit of the vaccine, and it gets kind of carried into the body through natural kind of suction or with some little bit of pressure there. And then it would get pushed into the uterus and then into the uterine horns. So we have done lots of studies to make sure that we select the vaccine formulation so it doesn't have an impact on the semen, which is important, right? Like if you develop a great immune response, but the animal doesn't get pregnant because the sperm is dead, that's a bit of a problem. So that was our first step, is to make sure that the formulation was not spermicidal. And so we've got that. So anything off the shelf that we could find was spermicidal, right? Because a lot of them have preservatives in there. And so, I mean, that's a non-starter. So we've been doing lots of playing with that. And we did lots of work where our first couple experiments, what we did was we took a parvovirus vaccine, and we just took the part of the vaccine. It was a commercial vaccine, and just took a bit of the inactivated parvo, and we formulated it with some adjuvants, and we administered it into the semen bag. But we used sows that had already been vaccinated. Like every parity, they got a parvovirus vaccine. The idea was, can we boost the immune response, right? And their responses were as good, arguably a tiny bit better than the intramuscular injection. So that was very encouraging. And then we started going into gilts and administering we did a little bit of work with parvo, and then we moved actually. The thing is, if you want to show that a vaccine works, you can show it gets an immune response, which is one thing, and which is, to be fair, when you often will stop because it's a little bit cheaper. But if you really want to show if a vaccine is working, you actually have to administer a challenge and show that they're protected against the disease. And we didn't happen to have a good parvovirus disease model in-house, but we did have a really good one for PED-V. So it's porcine epidemic diarrhea virus. The trick is that parvo is a reproductive disease. PED-V is more of a disease for the neonate. Like, it affects everybody, but the animals that are going to die are going to be the newborns. But if you administer the vaccine into the uterus, the vaccine response goes into the colostrum, and the suckling piglets do get protected. And then we could challenge the piglets and show protection. So we switched from parvo to PED-V. It was actually a bit timely too. So it was a couple of years ago, and that was really on everybody's mind. And we were just using that as our disease model. And then we're playing with all these different adjuvants, the dose, a little bit with the timing. So that was kind of neat. What we did was you're dealing with you've got a 2-year, 3-year grant cycle, right? So if you administer a vaccine at each parity, what are you at? 4 months by the time she's farrowed and weaned and ready to get pregnant again. And then if you're going to boost, then now we're looking at another 8 months. So time, if you're going to do 2 to 3 vaccine doses, you've gone through a year. So we kind of cheated a little bit, and I thought it was kind of clever. So what we did was we administered the vaccine with the semen into the gilts, but we purposefully gave her heat-killed sperm. And you could look at it under the microscope, and it wasn't like ruptured or anything, so you weren't getting some weird immune response. It was just they weren't swimming. And then 21 days later, she got pregnant again, or she came into heat again, right? So we could again vaccinate her again with the vaccine, and then heat-killed sperm again. 21 days later, we repeated, and we used the live sperm. And then, so that's a bit of a, I mean, that was a kind of a trial by fire, because these gilts had to cycle together, and they did in their gilts, right? So they're not all of them are gonna behave, right? So they all came back into heat properly. They everything was good. They had the litters. There was no difference in live, dead, in sizes, sexes, anything like that, or growth conditions. So it was all good. So what we learned from that is that we can do boosting in kind of a bit of an artificial setting, just to, like, that's not the idea. Obviously, we're not going to ask farmers to do that, right? Producers, but it was just to get the vaccine through a couple doses. And all the gilts had normal litter. So we were very encouraged by that. And the vaccine response, again, was good. Protection was okay. And okay is not good enough, but we were very encouraged. So what I mean by okay is like the piglets got challenged, and we do the challenge. We go into we actually moved to level 3 containment rooms at VIDO Intervac, because we just wanted to move to that level of security, and challenge them. And the piglets that suckled from the vaccinated gilts survived a little bit better and longer than the other ones. And then, of course, at the end of the trial, you have to euthanize everybody. So we were very encouraged by that. So then we have another round of funding where we are actually doing some very serious changes with our formulation. So we're actually formulating them into nanoparticles. And this is funded by the Agriculture Development Fund. And it's with Azita Haddadi's lab, and Ramin Mohammadi and George Mutwiri and our lab. So we've got these nanoparticles, which are very, very powerful. So you can take just a tiny bit of your vaccine, and for some reason, when you put it into a particle form, it gets taken up a lot better. I think your body might look at it like, are you a bacteria or a virus, something that's in a particle size instead of something just being fluid. And we get very, very nice immune responses to that. So the next thing is to do a proper trial and do a proper challenge experiment. So I think we've got 6 different forms of the vaccine, and we're testing them. Again, so it's just vaccine. So it's just a protein with these adjuvants that are in a particle form, and we're testing those at the Prairie Swine Center. Once we're happy with that, then we'll actually do the trial again at doing that VIDO in containment rooms and do the challenge. So hopefully within a year we should be testing those. So we're pretty excited.
Yeah, that is pretty exciting. It seems to be working, even if it's providing mild or I don't want to use the wrong
modest. It's not. Yeah, it's okay. It's just it can be better. That's all. No, we're pleased with what we're getting so far.
When you do that work, and this is my ignorance when it comes to this type of, can you do any measures on like how much of the antibodies are making it into the milk and transferring to the piglet, and like is that the rate limiting, or is there something else that's going on that
well, with PEDV, it's a good question. We actually are getting quite a bit of antibodies into the colostrum. We were quite pleased with that. The milk was just kind of okay, which was a bit surprising. I think we have to look into that a little bit more carefully. But what's happening is we're getting lots of antibodies, but they're not necessarily what we call neutralizing antibodies. So, I mean, it doesn't matter if you've got lots of antibodies, but they're not really doing very much. It's not very helpful. It's against the right thing, but for some reason. So what you want to do is have antibodies that would actually bind to the PEDV, so the actual virus, and stop it from acting like a virus. So you're actually neutralize it. And there's some really nice, very simple assays that you can run, and very clear. What you can see is it's like a clearing, you've got a cloudy little plate, and if you see a clearing, then you've got virus. If you don't see clearing, then that means the antibody's bound to the virus, and it's not acting like a virus anymore. So it's very much a night and day binary response. And And again, we're getting some neutralizing antibodies, but like our antibody response is really high. Our neutralizing is what's can be better. So we're learning, actually, I'm learning lots about the immunology of this. Like if you play with these different adjuvants, and we're keeping our route the same, right? Intrauterine, but play with the adjuvants, play with nanoparticle versus soluble. Is that really impacting your neutralizing antibody response? And I don't know yet, but we're very interested. So that's the trick. If we can find for viruses, if you can actually block the virus with these neutralizing antibodies, then that's great. Bacteria, it's a little bit different. Bacteria are funny. Sometimes an antibody will stop them. Sometimes they can decorate themselves with antibodies in a nice little coat, and they don't care. They still do what they're going to do. So, and that's where you need a cytokine response sometimes. So, but for a virus, yeah, we definitely know what we're looking for.
Yeah. So, you mentioned in your study design that you had, you did the killed sperm, and then you did it in order to like kind of make it so that you could do the multiples and see what that effect would. Would you expect the response to be different if you actually like did it over time, like you would have to, and had different ages of kilts or sows and that?
Actually, if it's about what it's 3 months, 3 weeks, 3 days, is that's the normal gestation, right? And then you've got some farrowing in there and about a week or so to get her back. So it's, when a rough guesstimate, it's about 4 months, right, between one breeding to the next breeding. That's actually a really nice time period for vaccination. It's very nice. So it's okay. I think the idea is that we would administer the vaccine at each parity, at each breeding. And I think what we would try to do is just administer it the first time. Okay, so usually you would breed a gilt at second estrus. Like first estrus would tell you, okay, she's sexually mature. Second estrus, then we administer the vaccine with the semen. And then if she gets bred the next day, the day after, like that is a bit variable, right? Sometimes you only give a gilt or sow even one dose, sometimes a couple. So we just administer it once the first time. And then, the next parity, give her the vaccine again. If we do our job to the best of our and anybody's ability, it would just be one and done. You vaccinate her once and then she'd have lifelong immunity. That's a pretty high bar. So right now we're just trying for every parity. But ideally it would be just once. And a little thing, little bit, I don't know how much people know this, but generally you need 2 immunizations to get at least 2 to get a really, really good immune response. I was actually amazed with these COVID vaccines at how after one dose they were doing, you had pretty decent antibodies. A lot of the times your antibody response is really low until you get your second one, and that's when it takes off. So I'm a little bit concerned about the gilts in that they might need a booster, because I want them to be protected in that whole first gestation. So we might have to think, like, again, if everything works wonderfully, yes, it would be one and done, but it's more likely we might have to give her maybe an injection 30 days into pregnancy, so it'll be intrauterine, and then wait a couple weeks, give her another one, and then after that everything is a boost. So again, it would just be once intrauterine. That's probably what's most likely is what's going to happen. And it's okay to mix them up. If you've got intrauterine and then intramuscular, like that's fine. You could do intrauterine, intranasal, however we get it to work best. But I'm a little bit worried about the gilts, that we're kind of asking a lot for this one little vaccine to fully protect them. But I'm not worried about the next farrowing. That would be a nice booster. And so any of the sows, I'm not worried about.
It makes sense. Maybe that's one good thing that came out of COVID is we're all a little bit more aware of what is required in vaccines and boosters, and times between those shots and how that
exactly,
impacts it. So I mean, it makes sense, but obviously the goal would be a one and done. That would be great. Producers would they definitely love that if they didn't have to go back to them. But and even if, like you said, if one can be the intrauterine and then maybe a booster is a different route, at least you've gotten rid of one, injection,
Yeah.
which,
Yeah,
helps quite a
exactly.
bit.
Yeah.
Yeah. Are there any, you mentioned PED and you started this with parvo. Are there particular viruses or, and I know you said bacteria is a different dragon altogether, right? But are there any particular that you are wanting to like target with this and look at?
I think I've got a bioinformatician in my lab, Dr. Zahed Katouni, who is working on figuring out what are the best targets on PRRS. So we actually are going to uh, use the adjuvants, the nanoparticles, and we're going to try doing with the PRRS antigens. And we'll do that just intramuscular and just some piglets just to see what kind of an immune response we do get. And if that's very encouraging, then we'll start writing some grants and start getting all the permissions required and move to complement the PED-V vaccine with a PRRS vaccine. So again, a really important reproductive disease in pigs. I don't see why you could really pick any target. Some of the interest that I've had is like with rotavirus or some of these pathogens that are really deadly to the newborns. This is all that is about passive immunization. So you're not protecting the piglets when they're developing in utero because that barrier is pretty thick, right, in the pigs. But all of a sudden they get born and you're expecting them to be protected and they're just not going to be. So what you do is you get the colostrum in the mom to have antibodies against rota or against strep suis or something. So it's more reproductive diseases, and then what diseases really impact the newborn babies is what we're thinking of for now.
I'll ask just because it's on everybody's mind lately, but would African swine fever be something that we would be looking at in that?
Oh, for sure. Like if we can get, yeah, I mean, it's just, for sure. Yeah, we've got people at VIDO who are working at the trick I understand with African swine fever is finding the target. It's been challenging. It's a big virus. There's lots of people been doing lots of work trying to narrow it down. And yes, if we can definitely find a really good target, then we would do that work. Again, that would be, I don't even know, that might even be, yeah, it's definitely level 3 kind of work. So it would be definitely under containment because it's not in Canada, thank goodness, and hopefully stays out. But yeah, for sure, if we can look at that one, that's not going to help the wild boars or anything like that because I'm not doing an intrauterine immunization any of the wild animals out there. But yeah, definitely the nanoparticle vaccine development. Lots of work going on.
Maybe you can work on something that they can eat.
Yes, exactly.
In the wild boar population, or blow dart gun, I think it might be.
Yeah, right.
A different way of doing it. Yeah. So I think it's like he's
Lots
all the
of
different
targets.
challenges, but yeah, because it's something that keeps popping up. But it definitely, I think a goal in the future. You wrote something in your little like about me thing that you said people may disagree, but I believe that with the right adjuvant and antigen combinations, subunit vaccines can work well for the industry. So I guess I'm just maybe speak a little bit more to that, and then maybe potentially why you think people may disagree with that.
I think it's well, the proof's in the pudding a little bit that I'm just trying to think. I don't know if there's any subunit vaccines. What I mean by subunit is you just take a certain protein, which has got the certain targets that the vaccine. So instead of having the whole PED-B virus, for example, that you kind of carve up, but all the pieces are there, that would be a killed PED-V or live attenuated. So all the pieces are there and it can kind of replicate a little bit, but just not very much. That's a live attenuated. Or you take some of the pieces, you formulate them in these nanoparticles. Most mucosal vaccines, so oral, intranasal, that are on the market are live attenuated. And it's just because there's lots of the pieces of the virus are there, or the bacteria, so there's lots of targets. The trick with live attenuated is you always worry a little bit about reversion to virulence. So it was live attenuated, and sorry, I can never remember if it's the Salk or the Sabin polio vaccine. One of them is live attenuated, and it's very, very good. But we basically have gotten rid of polio, except for some of the vaccinated people have been shedding just a little bit of polio, and you get these little outbreaks. It's really rare, but it does happen. So there's always that fear. The trick is if you can do just subunit. So say I take protein 1, 2, and 3 from the PED-V, and then I put in something really weird, protein X. And then we see animals in a barn and they have antibodies against protein 1, 2, and 3, but they don't have any antibodies against protein X. Then I can say, well, they didn't get those antibodies from the vaccine. There's probably a live exposure going on. So those are called DIVA vaccines. And so it's DIVA, differentiate between infected and vaccinated animals. So if you play with your subunits, you can add some proteins and take some out. The traditional form of DIVA is you actually take like the live attenuated PED-V and you take out a whole section. Maybe that's a bit of an aside, but I don't know. I think the proof is in the pudding. Yes, live attenuated, there has been lots of success, but I think the more we learn about adjuvants and targeting, I think if we keep playing with it and keep working at it, we can definitely find a vaccine that can get across the gut wall, uterine wall, whatever, and get impacted by the innate immune cells, and then help to direct an adaptive immune response. Like I think we just need to play with it a little bit, but you can't argue with a success. I mean, as far as I know, there aren't any subunit ones that are. There's lots that are in lab settings. We haven't made it fully to commercial development yet, but I think we're close on a lot of fronts. So,
okay, yeah, interesting. I mean, we've talked about it a little bit already, but I guess what would you be, or what would you say would be the next steps, or like a priority moving forward, either with your research or even vaccine research in general?
Mass delivery, especially for livestock, would be very, very nice. Anything where you don't have to say, okay, you need to get new barn workers or specially trained people. Anything where it's easy, like again through food or a nasal mist or in the water. Anything like that would be very, very well received, I think, by the industry. And see the next directions from there. I don't know. I think if we kind of think outside the box a little bit and try to think, okay, are there the more like I think it's important for the scientists to like for me, I didn't grow up around a pig barn. I had to learn the industry, and I'm still learning the industry. So it's important to kind of know what the husbandry is, so that we can change our approach to work with what is already being done. I can develop the best vaccine against almost anything, and I say, okay, guys, you got to round up your animals at this particular time point, and do it at a regular basis, because that's what the vaccine needs to work. Chances are it becomes a real money issue. Like, okay, is this worth doing? So it's better for us to learn what the barn does and work our vaccine design around the barn, and then the uptake would be, I believe, a lot better appreciated and hopefully used more successfully. So,
yeah, I think that's a great message for not just you guys, but every researcher out there, right? To understand what are you doing, because if it is too complicated or expensive to implement, then what's the point, you know? So I think that's great. So I guess our time is wrapping up, and so I'll just ask if you had like one take home message or conclusion for our audience. What would that be?
That I think that vaccines work, and it's if we can design them well enough and with a better price point, so that it can be affordable to do so. I hope that producers welcome vaccines, because I think it's really important to have, instead of getting in front of a disease that's already in the barns. If we can vaccinate, even if there's no disease X around, maybe not a bad idea to vaccinate, so you keep it out rather than fighting it if it does come into the area. So it's always a good idea.
Yeah, ounce of prevention and all of that.
Yeah, exactly.
Yeah. Okay. So our final three questions of the podcast are the same that we ask everybody. So they're a little bit different. And I think you were given this beforehand, but the first question is, what is your favorite swine-related book or resource?
I even have it here. I was ready for it. This is an awesome book. Can you see it? It's called Lesser Beasts, and it's all about the pig. And I have it as a hard copy, but I listened to it as an audiobook. And I was bailing of my husband's farm, and I could still, whenever I passed that field, with the last time I bailed, I can hear parts of the book. So anyway, it was really, really interesting to see the development of the pig domestication with the humans. So it was good. It's Lesser Beasts by Mark Essig, Essig.
I have not heard of that one, so I am definitely going to have to look that one up myself. So, yeah, no, that's interesting. So, our next question is, what is your favorite book or resource outside of agriculture?
I like the science books. I like Virolution, and there's the author's name is Jack Ryan. So he's really tough to look up because it always gets you to the Hunter Red October actor, and that's not it. He's the author, and his name is Jack Ryan, and he's got lots of science related books. He's very good. And I'm a sucker for the detective novel, that kind of stuff. In the summertime, I get a stack of just these crappy novels that I'm just quite happy to pour through in my backyard in the summertime. So,
and then our last question is, in your opinion, what sets successful swine professionals apart from those that are not? And you can define success however you want when you're answering.
I mean, good management, but also being okay with trying something new to see what are other people doing, what are other countries doing, and just exploring other people's success, and maybe even in other industries. I think all of that's knowledge is power. So it's okay to do things a little bit differently, within reason, of course, as long as you're doing all the biosafety and all that. But yeah, I mean, listen to your workers. A lot of people are getting workers from countries all over the world, and I kind of ask them, what are you guys doing, and how does it work for your people? So yeah, just listen and learn, and be open to new things.
Yeah, I think that's great. The resources are all around us. We just have to listen. I think it is a very good message. Well, Heather, that brings us to the end. So I hope you enjoyed your time here. I definitely enjoyed talking with you about this, and I hope the audience gets some good information out of this. So thank you very much for being on the show.
Good. Well, thank you very much. It was fun.